The International Prophecy Conference, an annual event which has become the prophetic conference in America, with this year's theme, Birth Pains of the Coming of the Lord. Packed services with attendees from across the country and overseas came to receive the latest prophetic insights and teaching from the world's most renowned prophecy scholars. Now go with us into this eye-opening, exciting message at the 2006 International Prophecy Conference.
The International Prophecy Conference. No other event like this one. And our next speaker is coming to minister to us, and I want to tell you a little bit about him. His name is Chuck Missler. Now, I got to say this, Chuck. I've read all these bios of all these speakers. Chuck, yours is 2 pages long. 2 pages long. There's more stuff that you've done than anybody else I can think of. Oh, he's going to come and help me.
I'm going to help you. You betcha. Mm-hmm.
Welcome to the International Prophecy Conference, my precious brother. He's brilliant. He's done an awful lot of stuff, and he's going to share God's word. Give him a big hand. Chuck Missler.
How you doing? Thank you. Thank you. Thank you. Appreciate that. That was wood hay stubble that we just threw there. But some of you are wondering why I wear these beads. I usually wear these just to make Joe nervous, you know. But actually, the real reason I wear them, or brought them, is I was in my workshop one weekend, and I inadvertently spilled a whole bunch of black and white beads on the floor. And as I picked them up, I thought, this is silly. I'll just put them on a piece of string. So I picked them up randomly and put them on a string and tied it so that now they won't spill again, right? But as I looked at it more closely, I was stunned because I noticed there were 2 white beads. Well, in Morse code, that happens to be an I. It is an I. Then there were 3 and 1, a dash and a dot. That's an N. That spells in, okay? 3 together, that's a T. There's an H and an E. In the B. It spells, in the beginning, God created the heaven and the earth. Genesis 1:1. It happened totally by accident. Why are you laughing? Do you think I would try to con you about something like that? Now you're laughing because you know in the pit of your stomach that that story is absurd. What you may not know, that in the field of mathematics and physics, it's defined as absurd. I want to start off by giving you what I call an epistemological IQ. Epistemology is a study of knowledge, its scope and limits. Don't waste your time taking it in college. It's given by the philosophy departments, and all they do is badger centuries of word definitions. They don't get to any practical result. But the question is, could this have happened by accident or not? Is it possible that my little story is believable, is creditable? Well, in other words, did it happen by chance, or was it a deliberate design? Now, some of you know that when I was 9, I was a radio ham, and Morse code is intrinsic, so it took me about 15 minutes to do this, okay? But what is the statistical probabilities? Well, it happens, there are 347 beads here. There's an alphabet of 2, black and white, right? Getting any single specific sequence of beads has 1 chance in 2 raised to the 347th power, which is the same thing as 10 with 104 zeros after it. That's a very big number. How big? Well, it turns out in physics, you can deal with probabilities that are asymptotic to 0. They approach but never get to 0. But mathematically, you need to have a place to cut them off, to have a cutoff point. And that's defined by Borel's law. In other words, any probability that is more rare than 10 with 50 zeros is defined as absurd. So that's a very big number. This is absurd to an absurd power, okay? Now, why am I getting into this? Because you and I are going to spend a little bit of time here reviewing biotechnology, and I'm going to try to condense. We have a 1 hour briefing on this whole thing, and obviously Joe wasn't too comfortable about my spending 2 hours with you, and so we're going to try to shoehorn it in just a brief summary. But why biotechnology? Well, the first reason, that's the reason for all our strategic trend monitoring, is it is prophetically relevant. But to me, that's, strangely enough, that's not the most exciting part to me. Yes, it is exciting, we'll get to that. Biotechnology offers one of the most profound epistemological discoveries of our day. In other words, as a proof of the reality of God. And you'll see why. Now, the other reason we're going to touch on this, we're not going to spend too much time on this, but it also is potentially one of the most dangerous technologies if it goes unbridled. And we're facing that very seriously. It's prophetically relevant. You know, Jesus made a remark in Matthew 24. He said, "For nation shall rise against nation, kingdom against kingdom, and there shall be famines, pestilences, and earthquakes in divers places. All these things are the beginning of" what? Birth pangs, right? Okay, got it. We're together. But the word pestilence is in there. Let's recognize that pestilences are prophetically relevant. In the book of Revelation, most of you that have studied the book of Revelation know it's got a heptadic structure, a sevenfold structure, 7 seals and 7 trumpets and 7 bowls. How many sevens are in the book of Revelation? Hundreds, probably 700 maybe. Some of them are very obvious, some of them are very hidden, but they're all there. But the first 4 of the seals you all know as the 4 horsemen, right? It's so well known, it's become an idiom in our literature. It's misapplied to a lot of things. But of those 4, you have a white horseman representing conquering, you have a red horse for war, and black horse, famine. And then the 4th horse is, in your King James, the pale horse, or more specifically in the Greek, it's the chloros horse. It's a pale green. It's sort of what you might call a seasick green, or maybe, what should I say, a mortuary green, but anyway. When he'd opened the 4th seal, I heard a voice of the 4th beast say, "Come and see." And I looked, and behold, a pale horse, or chloros horse, and his name that sat on him was Death, and hell followed with him, and power was given unto them over a 4th part of the earth. To kill with the sword, and with hunger, and with death, and with the what? Beasts of the earth. Now, you and I would jump to the conclusion that those beasts, we sort of visualize 4-footed animals or something. No, not necessarily. Some of those beasts are the kind of beasts you might search for with a microscope. Okay, let's understand that. And even Ezekiel has a similar passage. I said to the Lord God, "How much more when I send my four sword judgments upon Jerusalem, the sword, the famine, noisome beast, and the pestilence," and so forth. So again, we see that. So let's talk a little bit, first of all, very simplistically, about reemergent diseases and also deliberate diseases. Let's talk a little bit about global health on the dark side. About 50 years ago, with antibiotics and so forth, scientists predicted the end of death and suffering from infectious diseases. You remember those articles? Some of you are too young to remember, and some of you are too old to remember. But anyway, during the last 25 years, however, we've seen the reemergence and geographical spread of well-known diseases, including tuberculosis, cholera, malaria, a whole list of these. Except now they're more virulent than ever because they've become resistant to the antibiotics and so forth. So diseases once thought obsolete have become again a major threat. All kinds of new pathogens, more vicious than before. And there's also about 30 previously unknown diseases, like HIV and Ebola, for which there is no cure. The one disease we could talk about, not HIV being positive, but SIN positive. That's a genetic disease we all have. It also has a blood cure, but we'll get on. I'm sure you've heard about that. Okay, and so we can go through a real dismal litany of these things, and I won't go. There's very valid wars against these diseases, and we could talk much about them. But there's something else that I stumbled in doing the research here that startled me. Did you realize that there are little single-celled bacteria that take roll before attacking? It startled me. They call it quorum sensing. There's bacteria that when they're in your body anyway, but when they will take roll to see if is there enough of us ready here to have a chance at winning. If there's not, they don't attack. If there's enough, they call it quorum sensing. There are E. coli, Salmonella, Vibrio cholerae, and 30 others. This isn't just one or two. It's apparently a strange thing. And it sort of reminds you of Luke 14. You know, no king goes against another king unless he takes stock to see that he can win. If he can't win, he negotiates, right? Anyway, another fact I have to throw in here. Did you realize the number of physicians in the U.S. apparently are in the neighborhood of 700,000? The accidental deaths caused by physicians per year are about 120,000. So the accidental deaths per physician, according to the U.S. Department of Health Services, is about 0.171. The number of gun owners in the United States is about 80 million. And the accidental gun deaths per year is about 1,500. So that turns out, according to the FBI, so that says that doctors are 9,000 times as dangerous as gun owners. I thought that would be helpful for you. But I could show you the statistics about lawyers, but you'd be so shocked. I was afraid you'd be so shocked you'd seek medical attention. Now, this silly little business is obviously tongue-in-cheek. It's been echoing through the internet for several years. What's funny, you see a lot of rebuttals to this. None of them quarrel with the statistics. They just quarrel with the logic, strangely enough. But this is just done for fun. I'm just doing this it's a late evening, and I figured you needed something. So, okay. But I'd like to shift gears now, before we jump into biotech in another sense, and just indulge me what I'll call a tutorial perspective. I would like to take you on a tour of what I call a constellation of miracles that all occur inside a tiny city. If we were trying to build a map of reality, we might start with the human body, and then inside of which there are organs, and those organs are made up of tissues, and the tissues are made up of cells. The cells are composed of molecular robots, believe it or not, which below that you find atomic structure, subatomic particles, and non-localizable entities. It's interesting, as just a piece of background, that we discover that you and I are in a electrical simulation, a simulated world. The great discovery of 20th century science is that the universe is finite, not infinite. But also, the great discovery near the end of that century was that we're also in a digital world on the microcosm. It's finite. We're bound in finiteness. The world is not infinite, it's finite. On the small side, if I had a line and I cut it in half, I could take what's left and cut it in half again, couldn't I? I could take that half and cut it again. And you'd think, at least intellectually, I could do that forever. Turns out you can't. When you get 10 to the minus 35 centimeters, if you try to cut that in half, it ceases to have locality. It's everywhere at one instance. And that's true of length, mass, energy, and even time. There's not a unit of time smaller than 10 to the minus 43 seconds. You think, whatever it is, it could be smaller. No. That's why they call that field of study quantum physics. Everything's built up of indivisible units. But we're not going to go that far, but we just want to be sensitive to that reality. But we're just going to look at two areas. We're going to move down into the cells and molecular robot area. Now, Michael Denton, an evolutionist, to the best of my knowledge, not a believer, but he wrote a very landscape book called Evolution, a Theory in Crisis. In 1986, he said, one of his quotes is, although the tiniest bacterial cells are incredibly small, each is in effect a veritable micro-miniaturized factory containing thousands of exquisitely designed pieces of intricate molecular machinery made up of 100 billion atoms, far more complicated than any machine built by man, and absolutely without parallel in the non-living world. That's quite a statement, but you're going to discover before I'm through with that's a shocking understatement of the complexity. Darwin loved to talk about the simple cell, and our kids in school still get fed this nonsense. Well, the simple cell, we now discover it is unparalleled in complexity and adaptive design. Heavy trip if you're in engineering. It has a central memory bank. It is surrounded with assembly plants and processing units. It has repackaging and shipping centers, all made up of robots, protein molecules, typically 3,000 parts per each in three-dimensional configurations. And there are hundreds of thousands different kinds. And they have communication systems and language, error control procedures, and so forth. Let's assume that we were going to build a model of one of these things. And let's assume we're going to make a model 1,000 million times larger than life. Okay? Each atom will end up being about the size of a tennis ball. So far, so good. But we need 10 million atoms, 10 to the 13th, 10 with 13 zeros after it. If we counted one per minute, it would take us 19 million years just to count them. So I'm not proposing we really try this, okay? If we did, it would be over 10 miles in diameter. So you get a feeling we're not dealing with a simple cell. That's just the beginning of it. Now, it is obviously surrounded by a membrane. That's very glibly said, but that memory has gates that can tell enemies from friends. And when you don't know what the stuff's filled with, you give it a fancy name to pretend you know what it is. So that's the cytoplasm in there. But inside there's a nucleus. And that's the information center. That's the command and control center. That's the master library. Do you know which library in the world is read the most often? End to end? Yours. Inside your cell. And inside there's nucleus. There's automated factories and product manufacturing. There are power plants scattered throughout here to provide the energy for what's going on. You have apparatus for processing, packaging, shipping, and preparing for export. These are all functions going on in there. And there's also transport and trash disposal functions all going on in here. Now, there are robot machines, as I've mentioned. There's artificial languages and decoding systems. I'll talk about a couple of those. There are memory banks for information storage and elegant control systems, et cetera. And they also use prefab and modular construction with error, fail-safe, and proofreading devices. Let me back up a minute. Part of my career, I spent 6 years in the senior management of the Ford Motor Company. That was quite a few years ago. I was from 63 to 69, essentially right in that time period. And different company then, of course, for many reasons. But one of the proudest assets we had in those days was the largest integrated manufacturing plant in the world, the River Rouge plant. In those days, if you visited Detroit, you'd want to visit the Ford River Rouge plant. Why? There's a picture of it, an early picture of it. First of all, it started with ore docks, steel furnaces, coke ovens, rolling mills. They made their own, in the raw materials entered one plant, they made their own steel, glass, and paint under that roof. And it consists of 93 buildings with a tire making plant, stamping plant, engine casting plant. They had a part of a plant that automatically made all their engines. That part of the tour skip, it's boring. It's all this machinery, there's no one around. It makes all the engines. Radiator plant, tool and die plant, paper mill, even a soybean conversion plant. And that was all fed by a master power plant, 120 miles of conveyors, 100 miles of railroad, and 16 locomotives. Raw material, raw ore went in one end of the plant, and every 49 seconds a finished car came out the other end. That was what we mean by an integrated manufacturing plant, okay? Now, why am I going through this? Okay, we have this raw material coming in, new cars coming out. Get the picture. The simplest cell in your body is more complex than the Ford River Rouge plant. And it all happened by accident. Isn't that amazing? The simple cell in your body is not only more complex than the Ford River Rouge plant, it can do something the River Rouge plant can't do. Because in a few hours, it can replicate itself into another one. I don't think they ever tried to do that with the River Rouge plant. Okay, let's move on. Amino acids, there are hundreds of different amino acids, but it turns out there is a list of 20 that make up life. But it's even more complex than that in the sense that of those 20, as you can imagine, every complex shape can have a mirror image of itself, right? It turns out there are ways you can label the two images, the mirror image and so forth. One's left-handed, one's right-handed is the way they would describe what they call their chirality. Only left-handed ones are in life. There are some right-handed amino acids, they're toxic. Strychnine and others, okay? Most proteins are simply a long chain of amino acids, like a sentence, where the amino acids are like an alphabet. Some are hydrophobic, that is, they're insoluble water. Some are hydrophilic, they can dissolve in water. Here is a list of the 20 that turn out, just these 20 account for all life on the planet Earth. Just these 20. There are other amino acids, and we're also only talking the left-handed versions of these. Now, by the way, if life happened by accident, you'd have half of each. You'd have half left-handed, half left-handed, and you'd never get anywhere because they'd be toxic to one another. Interesting. Now, of these, half of them are not soluble in water. The others are soluble in water, just to have a characteristic of doing that. And the ones that are soluble in water, there's two kinds, those that have extra electrons, those that don't. In other words, some are acidic, some are basic. Doesn't matter. The point is they have certain chemical characteristics that turn out to be profoundly designed. If you've got 20 things like this, it's possible to designate a code using an alphabet of just 4. Doesn't matter what the 4 things are, but that's your alphabet. And if you take 3 out of 4, you can make a code that is error correcting for those 20 items. Now, there's probably not one engineer in 1,000 that knows how to go about creating an error correcting digital code. They know how to go to an error detecting code, do parity checks and so forth. But you can do parity checks, multiple ones, so that you can tell which bit is wrong. So there are ways to make that correctable. But unless you've had a course in digital design, you would not know what I'm talking about. But somebody had a course in digital design. Unless this all happened by accident, of course. The genetic code. Now, it also has punctuation. You've got an alphabet of 4, and you've got 3 out of 4 that you use to characterize whatever you try and do, including your start and stop, so you can parse your sentences. Terrific. Now, I'm not going to get into the chemical details, except there's 4 choices for our alphabet. And I'm just going to represent them by letters, A, T, G, C, all right? I won't embarrass all of us by mispronouncing these names, okay? But they're in pairs, and these pairs have an affinity for each other. So it turns out that if I take a string of these codes that represent some kind of sentence, each one will intrinsically attract its complement in pairs, which means that if I go ahead and split this into 2, each half will again attract its complement. So you realize this code suddenly is designed to be intrinsically self-replicating. Now that all happened by accident. Isn't that amazing? Now, what happens in your DNA, as you've all heard about, you know what DNA means, don't you? That's the symbol of the National Dyslexic Association, right? No? Okay, I'm sorry. I had to. It's your master record, but you don't want that to get out on the factory floor. So when somebody needs a copy of it, you transcribe it. You make a copy of it. That's called the RNA. And then it will be translated by some machinery to create the proteins that are implied by the codes. Are you with me so far? Okay. Now, the correct dogma was that you go from the DNA down to proteins. You can't go backwards. Wrong. HIV goes backwards and can skip generations and so forth. That's called the retrovirus. But anyway, move on here. What happens is you take the DNA and you make a 1-to-1 copy of it, okay? But turns out what the cell does, before it goes any further, it goes through and edits it. It takes sections of it out of there. Those are called introns. They're removed. And what's left are called extrons, and they're stitched together, and that gives you the thing that's going to end up generating the protein in a minute. Now, since I made up the slides a couple of years ago, what I also discovered, if you go to all the textbooks today in microbiology, they'll call these introns that are removed junk DNA. That's the term they'll use. because they concluded that since they don't have anything to do with protein generation, they assumed that they must have some function that's long been lost in evolution or something. So they're called junk DNA, and they have been discarded till a few months ago. If you read the Scientific American, you know there's some interesting articles because they discovered that even though they don't generate protein, they seem to have some other functions, architectural functions. Imagine that. But here's the part that blew me away, not that. The thing I picked up from that article, do you know what percent of the DNA is considered junk DNA? 95%. All this talk you hear about, we've uncovered the human genome, is a 5% sample. Now that's okay, but you'd think they'd have a little humility to acknowledge that they have just scratched the surface. They don't begin to understand all the complexities that this design includes. Now, what happens to this messenger RNA is there are, if you imagine them like recording heads that move along, and they move along, and they have a start, they come to a start bit, and then they take the next code, which is one of the 20 amino acids, and then the next one, which is another amino acid, and they concatenate these, they take the codes, interpret them, connect that amino acid, and make a chain of amino acids. And that chain becomes a protein. And we're not talking a dozen, we're talking 500, and others, they're significant lengths, obviously, and as it goes along. So now, and here I'll give you an example. Each of these amino acids are a fairly complex hydrocarbon, carbons and hydrogen atoms. And I won't get into that. Each one is a little different. Each one has different properties. Some are soluble in water, some are not, some are acid, some are basic. But they all have a group that has the characteristics of becoming a chain. So these things intrinsically will chain themselves. I shouldn't say intrinsically, the other proteins will hook them up. But the point is, you end up with a chain of these things, let's say 500-600 long. Get the picture? Then when you get to a stop bit, it's cut off. Because of the water, some are soluble, some are not, they'll take a shape. The positive sides will attract the negative sides. They will take a three-dimensional shape and become a robot that has a function. Now, the negatively charged groups with the positive charged groups, the hydrophobic side chains stack in the center, the hydrophilic go to the outside, and all that ends up with a minimum energy conformation, which becomes a specific robot of some kind. Now, what I want to tune you into something here, I want you to be sensitive to two different kinds of technology. There's the technology of conveyance, like radio or television. Those are media, if you will, in contrast to the technology of content. Our problem with television, they've got incredible technology to make it high definition, but they've got nothing worthwhile to transmit over it, okay? But take for an example, I want you to distinguish between the media and the manuscript. If you talk about books, you can talk about technology, bindings, paper, ink. That's a technology of bookmaking. It says nothing about what the manuscript has to say. Do you follow me? There's two totally different. Now, the frontier in science, in fact, the frontier in every field of science is now turning out to be not chemistry, not physics, make a whole list, is what we call the information sciences. There's a whole field of study called the information sciences. And I come from that background. That's probably why my Bible studies are a little weird, okay? Now, I gave you this little example of 347 beads from an alphabet of 2. I want to now introduce you to 574 elements long out of an alphabet of 20. This is being listed here as the hemoglobin molecule in your blood. 574 amino acids long. There's the list. You need 36 of those, 68 of those, et cetera. And they have to be essentially in a certain sequence. What's the probability of getting a successful one? Well, there's a formula from switching theory that'll give you that calculation, and I won't take you through that. This alphabet of 2 and 347 turn out to be 10 with 104 zeros after it. The hemoglobin molecule, 574 elements out of an alphabet of 20, is 10 with 650 zeros after it. Now, you will laugh at me out of this building trying to sell you the idea that this happened by chance. And yet you let your kids go to school where they cram down their throat that the hemoglobin model just happened by accident. Can you imagine? The ancient pagan cultures used to carve out of wood, stone, or whatever and worship gods that are insulting. We've invented a more insulting God than they did. We have ascribed the majesty of the creation to nothingness, to the randomness of a roulette wheel, in effect. Can you imagine? Anyway, enough of that. Let's keep moving here. Just to give you, these numbers are big numbers. 10 to the 18th seconds is all this occurred. If you take 15 billion years, I don't, but some people do. I was on a board with Dr. Teller and the sidekick Norris Keeler, and they were talking about 10 to the 18th in one of the discussions. I said, gee, that's longer, that's older than, that's more than 10, there were seconds in the universe. And they looked at me shocked. What do you mean? You know, at Livermore and so forth, they use these kinds of numbers all the time, but it hadn't occurred to them to connect the dots, so to speak. If you take 15 billion years, which would be their presumption of the age of the universe, that's only 10 to the 18th seconds. See, these numbers are bigger than you realize. They're competent estimates that there's only 10 to the 66th atoms in our galaxy, total. 10 to the 80th particles. These are big numbers. So when you start getting to the 10 to the 50 or the 10 to the 100 or 10 to the 60, you're talking way out of reality in terms of probabilities. In fact, they're defined as absurd. The idea of hemoglobin molecule happening by some accident of chemistry is equivalent to winning the lottery. Not one day or 2 days in a row. How many think you would have any chance to win the lottery 2 days in a row? 3 days in a row? How about 90 days in a row? That's the probability. Your acceptance that hemoglobin is by accident is your equivalent. It was like winning the lottery. And that's just one, of course, one end. We could go on and on. But let me tell you, how many in here are fishermen? Okay. I want you to imagine an experiment where we're going to take two monofilament lines to represent DNA. They're going to be 125 miles long. And I want you to design a way that you can take that pair of monofilament lines and store them inside a basketball in such a way that you can take it out, split it apart, copy it, and put it back without tangling. 120 miles long, at 3 times the speed of an airplane propeller. That's what's going on in your body. There's an incredibly clever, elaborate set of spools and stuff that accomplish that equivalent thing. Just the mechanics of handling it is out of sight. So which came first, the DNA or the proteins? You can't have proteins without DNA, you can't have DNA without the proteins. See, the problem is that it takes protein, each requires the other. Which means, by the way, they each had to be created at the same instant. See, thoughts and language are not physical. There's a whole other technology. Darwinians cannot explain the origin of life because they cannot explain the origin of the information. See, it's not the technology of conveyance, it's the information. Now, this makes sense except in one place in the world where it's against the law to think, and that's the U.S. You cannot teach intelligent design. Forget creation for the moment. Forget these other issues. What we should be pushing for is evidence-based education. But they won't let us. It's against the law to teach or even hint at intelligent design. It has to be protected by the law because it's totally irrational. Psalm 19, you all know it. You've seen it, heard it many times. The heavens declare the glory of God. The firmament shows his handiwork. Day unto day uttereth speech, and night unto night uttereth knowledge. There is no speech or language where their voice is not heard. Their line has gone throughout all the earth, and their words to the end of the world. Have you ever noticed the vocabulary here? They declare, they uttereth speech. They showeth knowledge, speech and language. This is all in information science. It's not astronomy. It's information sciences. That's the language, that's the frontier in microbiology, and that's the final nails in Darwin's coffin. Anyway, obviously you can tell digital codes require pre-planning. We get into the whole design. Anybody's been in a design team knows you better have coordination. You can't have these guys doing asynchronously random anything. You got to keep it integrated if it's going to work when you're through, and we could spend some time on that, but I'll spare you that. Talk about a little bit. Let's go outside the cell. We have plants and animals, and they are, interestingly, dependent on one another. Now that's curious. In the fall, we see the leaves change color because they're losing their pigmentation. There are two particular pigments in the green leaves that you depend on. You take the leaf apart, and you're again confronted with the most complex designs, with pipelines carrying stuff from one place to the other. And we could go on and on about that. But and I won't take you through the Calvin cycle. Those of you that know biology, relax. I'm not going to go down that path, except to say that there's two separate processes that depend on two different pigments called P700 and P680, which are the frequencies of green that cause the right behavior to create electron transfer chains with these two pigments, and a whole bunch of enzymes which do three things. They fix the carbon, they fabricate glucose, and they deal with aerobic respiration. For us, there's just a big box called a leaf. Let's do a black box approach here. Light shines in here to provide the energy for this all to happen in two different frequencies, and it needs CO2 for a raw material, and it needs water coming up from the ground, the roots, what have you. And out of this process comes a complex molecule we would call glucose or sugar, okay? And it turns out, we don't have to go into the particular molecule, that's not critical for our purpose here, except it might turn, if you're adding up the arithmetic, you'll know you need 6 CO2s, 6 waters to get one sugar molecule and 6 oxygens out, okay? But it's all balanced, it all works. The complexity inside the leaf is unbelievable. I'm sparing you all of that. But here's the point. Photosynthesis means with light, I don't think we need to spend, we have 170 billion metric tons of extra carbohydrates. It makes more sugar than the plants needs. That's where we get our food, and so forth. Anyway, the net of it is, the plants give off O2 and sugar for what we call food and oxygen. The animals, of course, us, need both of those and give off CO2, without which the plants can't survive. See, what's fascinating is this all happened by accident. I love this when the ecologists say, boy, if we have a 0.5% change in the ozone layer, it's going to be cosmic doom. The next time someone tells you that, say, gee, that's fascinating. Who balanced it? That argument that is so delicately balanced is an argument that it couldn't be accidental. Somebody went through a lot of trouble of tuning it. Get the point? Turn those coins over to the other side of it. Paul Davies, not a believer, but a brilliant mind, points out, says, it seems as though somebody has fine-tuned nature's numbers to make the universe. The impression of design is overwhelming. Does he know he's breaking the law? He's teaching design. There's a term by secular scientists called the anthropic principle. What they mean by that is, as they study the universe and put together everything we think we know about it, we discover that there are all kinds of relationships, ratios, that are delicate. That if they're not exactly the way they are, life is impossible. If the Earth's a little closer to the sun or a little further, it's either too hot or too cold. If it turns a little faster or a little slower. There are hundreds of these observations, some of which, if you change them 1 part in 10,000, that little, life is impossible. Psalm 39 summarizes it. Thou hast possessed my reins, thou hast covered me in my mother's womb. I will praise thee, for I am what? Fearfully and wonderfully made. Marvelous are thy works, that my soul knoweth right well. Thine eyes did see my substance, yet being incomplete. And in thy book all my members were written, which in continuance were fashioned, when as yet there was none of them. It's all forethought. God planned you, every one of you individually. And my information science friends that know a little bit about biology will point out something else to me that I just have to throw in here. Let's assume that every one of you in this audience could play any instrument in the orchestra. You had the skills. And let's assume that all through here we're littered all the musical instruments you might want to use. And let's assume I handed each one of you a complete copy of the symphony, the orchestration, the whole thing. Each one of you has a copy of that. Would we have a symphony? No, because somebody has to say, you're going to be percussion, you're going to be first violin, assign something. And there's a process in computer architecture called conflict resolution logic. Somebody has to be the quarterback, right? You take some cells and they divide identically. But then as they continue to divide identically, you look, they're no longer identical. You start having a backbone, you start having muscle, cortical. That's being orchestrated. An information scientist will point out to you there's a part of the equation that's being overlooked, and that is it requires external input. I believe God is personally involved in every cell division of your body. That's hard. We can't possibly grasp that, but I believe it's true. Well, let's talk about the new Sorcerer's Apprentice. We're tampering with the engines of creation. Now, Sorcerer's Apprentice, you obviously, von Goethe did a thing called the Magic Student in German, and that became a musical piece by Dukas in 1897 called The Sorcerer's Apprentice, and of course, most of us have been exposed to that, if no other way, by Walt Disney using it in Fantasia back in 1940. Again, some of you are too young to remember, some too old to remember it, that's all right. But the whole theme of the play and the music and the Fantasia is the idea that a student of being a sorcerer cast a spell that wasn't quite correct. And that unleashed forces that were uncontrollable, that his teacher had to intervene and to keep it from becoming a total disaster. And that's why I like the term Sorcerer's Apprentice for microbiology, because that's the same thing we've got going on here. If psychokinesis results in identical cells, why do they differentiate? You know, if we watch two cells, they split, and then those two split to become 4, and the 4 become 8, and so forth. That becomes, ultimately, it grows into what? An embryo. Who's quarterbacking that? Before they differentiate, they're called what? Stem cells. You've heard that term in the press probably. I want to straighten out some, and I won't get through the Carnegie stages, that you get the idea. A stem cell is made from there's a code of 4, 20 amino acids, over 100,000 prototypes that make up the elements in the stem cell. But eventually those stem cells differentiate to become cortical tissue, muscle tissue, bone tissue, whatever, okay? Several hundred different kinds of cells that become tissues, organs, up to the 10 to the 13th cells that make up your body. Now, what comes along here is a concept of nuclear transfer, where we interrupt that process. When it's still a stem cell, we remove the nucleus and put in the nucleus of a donor. And if it happens the way you're hoping it happens, you end up with a replica of the donor. It becomes a replica of the donor. We call it cloning. Your technical term is nuclear transfer. And now what makes this, we've all heard about the clone in sheep's clothing called Dolly, all made possible by the technology of a needle-making machine that can remove and insert these tiny, tiny nucleus of a cell. And they remove that, and out of all that business you get finally an actual sheep from a surrogate mother. She was cloned and so forth. And I should point out right away, that was after 274 failures. So don't volunteer for any cloning experiments. And today it's very common. You've got monkeys and pigs and all kinds of creatures that are cloned. It's very common today. In fact, just a few weeks ago in Taiwan, they cloned pigs with fluorescent that glow in the dark. Even the innards glow in the dark. Now, I don't know what good that is, but it's curious. I don't know if you can tell that on the slide or not, but anyway, they glow. Anyway, there are a lot of questions. When and why do replicating cells no longer remain identical? And this leads into the whole field of genetic engineering, manipulating stem cells to produce desired tissues. That's only one part of it. And nuclear transfer to replicate complete beings, cloning. There are all kinds of moral, ethical, legal, and theological issues here that we don't have to spend time on. Just be aware that you're really dealing in some thorny areas here. When does tampering with an embryo become murder? That's an issue. You know, the minister and the priest and the rabbi were arguing about when life begins. The minister said life begins at birth, and the Catholic priest says, you are really out of date. Life begins at conception. They argued about that for a little while until the rabbi says, you both have it wrong. Life begins when the dog dies and the kids are off to college, But seriously, is it acceptable to clone a baby to save a child's life? That happens in Colorado. Molly Nash had leukemia. They in vitro developed embryos that created the necessary cells to keep her from leukemia, and she's doing great. But what, is it acceptable? If it was your daughter, you'd do it. Yes, absolutely. And of course, we get the whole business of life beginning. When somebody wants to talk about life beginning, remember this fact. When did John the Baptist begin his ministry? When he was 9 inches long, weighed 1.5 pounds, he jumped for joy, was spirit-filled. Can a human clone be saved? Scientists tell us it's just a matter of time before we have clones walking around our society. Okay, can they be saved? I don't happen to think so, because they're man-made. But it raises some very provocative questions, which you really won't understand a major part of the Old Testament until you understand what a Rephaim is and what a Nephilim is out of Genesis 6 and so forth. But, and what are the prophetic implications? Jesus said, as the days of Noah were, so shall the coming of the Son of Man be. What do you mean by that? Don't understand that? You better know what happened in Genesis. What were the days of Noah like? And boy, that's an interesting study for another evening. Well, Woo-Suk Hwang, South Korean scientist, became a national hero not long ago. In May of 2005, he and his team announced that they had successfully cloned a human embryo for scientific research. Made the news. In August, he announced that he successfully cloned a dog, an Afghan hound, named Snuppy, and so forth. All the news, a lot of brouhaha there. Then in November of 2005, one of his key researchers ceased collaboration. And by December 23rd, a official probe discovered that at least 9 of the 11 stem cell lines were faked. Hwang intentionally fabricated stem cell research results, and it's caused a whole echo throughout the technical community about, they thought science was a peer-reviewed journal. No, it's highly respected, but it's not a peer-reviewed journal. But the mainstream press is so intoxicated with embryonic research, they still won't acknowledge that adult and umbilical cord stem cells are really the successful avenue to go for. In California, a deceptive $35 million campaign convinced California voters to pass Proposition 71 last year. It authorized the state to borrow $3 billion to subsidize research in somatic cell nuclear transfer, cloning. It makes you wonder, I thought California had these highly venerated venture capitalists. Were they asleep at the switch? I don't think so. They're smarter than that. Because it's not $3 billion, by the way, it's $6 billion, because the interest over 30 years adds up to the same amount. So you got, here's California with enough debt already that's been conned into this whole mess. And we go through all the problematic thing here. Heck with that. Let's just move along here. There are two different kinds. Something you will get confused by the way the media presents this stuff. They deliberately confuse you. There are two kinds of stem cells. There are embryonic stem cells that could be used for cloning, and they can also be used for reproductive cloning for other medical reasons and for therapeutic cloning. Then there's another group of cells that are adult stem cells that can be taken from all kinds of places, extracted from fatty tissue, blood, bone marrow, and elsewhere. The first are very controversial and never have succeeded producing any results. The other one is very easy to do, highly reliable, has a long history of successes. And the adult, or that is post-birth stem cells, are taken from the umbilical cord, blood, bone marrow, extracted from so forth. And usually from the patient's own body, so there's no problem of rejection and so forth, typically, not always, but usually. And there's no mutation issues and so forth. And they typically generate the cell from which they were taken, but they also have found ways to make them do other things. They have blood cells becoming neurons, liver cells that can be made to produce insulin, but there's a number of things going on, and this is actually very exciting. Give you some quick examples. 26 paralysis patients, these were paralysis patients, were able to regain sensation in the lower body, bladder control, and even arm and leg movement from spinal cord transplants. Some paralysis patients were able to walk less than a year after having the surgery. There's also heart disease and cardiac degeneration examples, corneal degeneration. 20 patients treated in recent studies, 16 either regained their sight or experienced increased levels of vision. The real point I'm getting at is There's all kinds of places that they're optimistic about making changes. Let's take a look at the scoreboard. This is the part I want you to remember. Embryonic stem cells. How many successes? 0. No track record of anything working out so far. Adult stem cells, thousands. Gordon, who's a basketball player, his attitude to this is, that's hardly a game worth watching. Well, the myths of yesterdays tend to become the reality of tomorrow. The price of unbridled curiosity. Somebody hands you a box that says, do not open it under any circumstances. And then you time yourself to see how long you can stand that before opening it. That's called Pandora. In Greek mythology, she was the first woman on the earth, and the gods had given her a box, warning her never to open it. Now, I don't think there's any guy in this room that's stupid enough to expect that to be followed, right? So the Greek gods weren't too bright, in my opinion. And obviously, as the myth goes, she opened it, and out of it came innumerable plagues for the body and terrors for the mind, the Pandora's box syndrome. That's exactly what we've got here. H.G. Wells, in 1896, wrote just a fictional novel of the island of Dr. Moreau. And in his novel, he has all kinds of animal-human hybrids that populate it. In today's laboratories, hybrid animals are called chimeras, after the Greek mythology, a mythical Greek creature with a lion's head and a goat's body and a serpent's tail. Scientists have created sheep that possess human hearts and livers. Now, from a research point of view, that's quite exciting, but that, if you start thinking it through, is terrifying, because you're starting to create diseases that cross species that we have never seen before. Pigs have been born with human blood. Very interesting if you're researching blood, but this is not going on in well-organized government laboratories or large corporate procedural laboratories, in garage startups, thousands of them around the world, with no time or energy spent to safety in a race for patents and breakthroughs. They're altering the genetic makeup of the creatures on the planet Earth in ways that we cannot anticipate, which can mutate in directions we don't have any idea. Of mice and men, at the Salk Institute in San Diego, they recently engineered mice that possess a small percentage of human brain cells. I think I've hired a few of those. Scientists injected human embryonic stem cells into the brains of rodent fetuses, resulting in the birth of mice with both human and rodent brain cells. And of course, what they're trying to do is create models that can be useful in studying neurologic disorders, Parkinson's disease and Lou Gehrig's. The motivations are very high, don't misunderstand me. But the possibility of adverse surprise is disturbing. At Stanford University, they injected neural stem cells in mouse fetuses. They also made the first mouse with a nearly complete human immune system. That scares me the most. There are few safeguards against errors or abuse. Cross-species implications, we want to talk about that. When you start messing around with these kinds of things, you're opening Pandora's box, in effect, for unknown diseases or complications. And you have the potential for self-replicating mutations, and they're impossible to fully anticipate. And of course, there are very few regulatory or procedural disciplines. Now, there's a European think tank in Stockholm that said the free access to genetic sequence data for the human genome and a large number of other genomes, including pathogenic microorganisms, could pose a significant threat if misused. Terrorists can take this and start to make diseases that are targeting individuals, certainly ethnic groups. That's really the point that the CDC and others talk about. You may be familiar, maybe not, with what happened in 1918. There was a pandemic they called it the Spanish flu. And a virus, in a matter of about 4 months, killed almost 50 million people in 4 months. It came, wiped out somewhere between 20 and 50 million people, and then disappeared. This is more than the Black Plague that we read about in the history books. It's worse than AIDS, but it disappeared. After a 10-year study, they've been trying to find out what caused it. They spent 10 years digging up bodies from back in 1918 and trying to get DNA and stuff to analyze to figure out what on earth this was. And they have found out what it was. They took lung tissue from 2 soldiers and an Alaskan woman, all of which that died in 1918. It apparently was a bird flu that jumped directly to humans. And there's reason to believe that there are pigs involved. Where you've got poultry and pigs together, you have a high degree of risk. And that's very prevalent in China, which is one reason China's got all these problems. And I just found out yesterday there are now 94 deaths in Europe to the avian flu, H5N3. The genetic changes cause this thing to become lethal. And so the World Health Organization has attributed at least 94 that they're watching from human. And so we could go on and on about this. You can skip generations. You can also create directable intruders. You're starting to see fiction stories build plot lines on these kinds of things. But I want to talk to you a little bit about nanotechnology. That's the technology that involves the manufacture and manipulation of materials at the molecular level. I happen to be working with a company that's one of the experts in that nanopower technology. And these are small things. They've actually made a gyro that's on chip. It's microscopic, but it's a gyro, so the chip knows where it is, you know. So this nano is just the billionth of a meter, that's where we get the name. A sheet of paper is about 100,000 nanometers thick. So nanometer, just a way of something says very, very small. And they could have enormous effects on things that might go for a number of areas. And there are at least 80 consumer products already using these technologies. So there's nanotechnology. It has as its focus molecule-sized machines. Okay. There's also a field of study called robotics, which has as its goal self-modifying sentient machines. What do I mean by a sentient machine that can simulate cognitive behavior? Robot. That's their goal. There's another field of study called genetics, and their goal is the self-replication of manipulated entities. Now, what's happening, as you can obviously see, all 3 of these are converging. Nanotechnology and robotics and genetics are beginning to attend the same conferences, so to speak. One of the goals would be to create self-replicating, sentient machines capable of directable diseases targeting specific groups or individuals. You could make a disease that targets only people of a certain ethnic origin. What would happen if Joseph Stalin or Adolf Hitler had access to that technology? Boggles the mind. Well, bioterrorism, here we go into a whole litany of examples where this is going on. Now, biological weapons are a terrible military weapon. They're very difficult to control. They're not very effective, but they are the dream weapon of a terrorist. Why? Because the symptoms will take several days to show up. They can insert into a culture and then get out of there before it is even recognized. And if it's contagious, as many of them are, it'll spread through the whole culture. One person gets on an airplane can affect the entire plane on a coast-to-coast flight. And treatments are elusive and uncertain at best. And inoculation is always going to be limited to some extent. And the logistics of limited supplies, triage, and all that sort of thing is always impossible to resolve. And now there's the briefing package we have. I'm not going to get into it. There's a whole DVD attached to it of the CDC going through a history of biological warfare. I'll spare you all that. I really want to shift gears here and get into the biblical implications. If you haven't done a study of the Nephilim in Genesis 6, you enter that, and it's a very controversial area because most seminaries have mistaught the passages from the Scripture. If you get into Genesis 6, recognize the first 2 verses are a single sentence. And there's something very spooky that went on, where Satan attempted to create hybrids called the Nephilim, the fallen ones. And but that's the only part of the story. The flood wiped them out. That's why he sent the flood. But it also happened again later. When Moses sent in the spies, the 12 spies, 10 of them came back terrified because Numbers 13:33 say there were Nephilim in the land. So we need to understand why was Joshua told to wipe out every man, woman, and child of certain tribes, because they had a gene pool problem. So you need to get into that. Well, the other thing we could talk about is the potential role of clones. And in Daniel 2:43, we have the iron mixed with, remember the famous image of Nebuchadnezzar. They had the iron mixed with clay. I've taught that passage for decades, never reading carefully verse 43, where Daniel explains the miry clay. It's in Aramaic, not Hebrew. There's a little tricky there. But the point is, it says As for the miry clay, they, he switched to a personal pronoun, shall mingle themselves with the seed of men. Now, I had my Jewish exegetes confirm the grammar. In order that means the they are not of the seed. You can't mix if they mixed with the seed of men, which means they are not the seed of men. So that's not the only explanation, but one thing it suggests is some participation of hybrids of some kind. Revelation 17:11 is a classic conundrum among prophecy scholars. And I'll just leave you to read that and make sense of it and try to make sense of it without cloning. I can and do, frankly, when I teach it, but I'll just leave you to chew on that because it could be an illusion. But you can't get into this subject without somebody asking about the mark of the beast. Don Perkins did a wonderful job earlier today dealing with that. Revelation 13:8. People who know nothing else about prophecy all know about the 666. Let's just touch on that for a little bit because there are a lot of misunderstandings. In the Greek, the word Christos, you take the first and last letter and put the serpentine character in between, and you've got the expression for the Antichrist, or more precisely, the pseudo-Christ, and those happen to have a geometrical value of 666. Fine. But whose number? Everybody gets confused. Your number is not the problem. It's not your PIN number or credit card number. That may be the mechanism by which he has control, but that's not the problem. The problem is his number. See, a lot of people point to barcodes and insertable chips and all that sort of thing, and because some barcodes use a 6 as a separator, and we have these insertable chips that are used for inventory control. They have now coming the RFID chips, where you can walk through a warehouse, and you know what's in the warehouse by walking through it, in effect. And that's all coming in spades. And what they overlook, though, people get into that, it's his number we're talking about. And the point is, it's an identity that somebody takes to identify with him. You're not doing that when you get a credit card or something. No. You're not. If you're on special ops in the military and you get an insertable chip so they can track you in satellite, that's not what we're talking about. That's not what we're talking about. I think there's only one physical description I know of in the Bible of the Antichrist. It's the last verse of Zechariah 11. Woe to the idle shepherd that leaveth the flock. The sword shall be upon his arm and upon his right eye. His arm shall be clean dried up, and his right eye shall be utterly darkened. We presume, it's a conjecture, but we presume that might be an after effect of the head wound that becomes so prominent in the Scripture. And what seems to be the suggestion here is people who are agreeing to swear an allegiance to him take an insignia to represent that publicly on their hand or on their forehead. And obviously his ability to enforce that is his power on the electronic fund system, whatever. But it's his identity, and you and I don't have to worry about that because if you're saved, you won't even be there. That's after the rapture. 2 Thessalonians 2 makes that very, very clear. If you're not saved and you think you're going to go through that after the rapture, you got a serious problem. Because the one problem that Tim LaHaye and I have, the whole Left Behind series presumes you've got a second chance. And 2 Thessalonians chapter 2, verse 9 leaves a little problem. If you rejected Jesus Christ, God's going to give you over to a delusion. Scary stuff. You don't want to play that game. But let me tell you about a technology. I love to throw something in here you probably haven't heard at some other prophecy conference. Joe generally wants me to do something here that you carry away that's unique, right? Okay, so I'm going to call this Reach Out and Touch Someone. I want you to imagine this. Before you leave a business-related luncheon, you shake hands with 5 different colleagues, collecting their electronic business card information by a simple handshake. Really? How are you going to do that? You walk out to your parking garage where your car unlocks as you touch the door handle. It knows who you are. You pick up your son from school. When he hops in the car, he sticks a wireless headset in your ear and says, "Hey, you got to listen to track 3, Dad." This is what's called red tacton technology. Through the newest advances in photonics and optics, electronic data can now be sent over the surface of your body, not on your skin, above it. There's a magnetic field around your body, and that can be used to transfer information. The human skin has electromagnetic fields over which electronic information can pass. Red tacton transceivers can electro-optically use these fields to send data at about 10 megabits per second over the surface of the body. This is an announcement just recently by Nippon Telegraph and Telephone Corporation. And they're leading on this, but there'll be other companies doing the same thing. And so you drop your MP3 player or your PDA or your cell phone in your pocket without missing a beat, literally, because the current never touches the human body itself, only its magnetic fields. And you don't have to take your ID out of your, when you're walking with your PDA, your pocket. It being in your pocket's enough identification. And so if you're dancing with a partner, you can, your MP3 player can be transmitted to both of you while you hold hands. Kind of cool. See, the devices don't actually have to touch your skin. So it provides a way to pay bills, to gain access to secure locations, that sort of thing. Well, that's just a little throwaway. You might be on the alert for that. But the real point tonight is man is invading a world which he has only the faintest glimpses. And the uncharted impacts can replicate unseen, transcend borders, species, and even generations. Now, only the most committed can persist in denying the role of a creator. And by the way, I have to lay this on you. It shocked me. I've taught the book of Romans for many years, obviously. Only in the last year or so did I pay attention to what Romans 1 was really saying. When we were doing our Genesis commentary, which is now being circulated at the National Laboratories, incidentally, it really startled me as I was doing some background for that to realize how throughout the whole Bible, God is jealous of his role as creator. As Christians, we tend to focus on the redemption, death of Christ, and all that, and that's appropriate. But before all that, just the it's interesting throughout the Bible how God underscores his concern to be recognized as creator. In fact, in Romans 1, he pronounces on a culture a specific judgment for those that refuse to recognize him as the creator. Do you know what that judgment is? Read Romans 1 from verse 20 on. I'll tell you what it is. Homosexuality. I've always looked at homosexuality as a sin from Leviticus and all the passages we always quote. Now let's not get confused. God loves the homosexual. He hates homosexuality. He loves the sinner. He hates the sin. We get that backwards. Let's be careful about that because Christ died for them. So I don't want to confuse the fact that homosexuality is a choice and it's a sin. But God says for them not recognize him as creator, God says, I will give them over to. And he goes on verse after verse, making it very, very clear what he's talking about. And it stunned me to stand back and realize that the sin of Sodom and Gomorrah was not homosexuality. It was the public condoning of it that caused them to be destroyed. Yeah, well, I was right. And it stunned me to really stand back from that text. A text that was so familiar, it's probably so familiar to all of us, and yet realize what he's really saying. It's very disturbing. Yes, we are indeed fearfully and wonderfully made. Now, there are topics I did not get into, the chimera I didn't get into, agricultural genetic manipulation, huge, huge scandals, billions of dollars of lawsuits from these companies that got messed up with corn and all. I won't go through that here. The fact that we're having genetic athletes starting to surface, that's very hard to test for. The biblical precedents, prophetic implications we just touched on. Now we, as I say, we monitor a dozen strategic trends: weapons of mass destruction, rise of Islam, the rise of Europe, the rise of Asia. We'll talk about that tomorrow. Struggle for Jerusalem, and so on. And the challenge to America. I'm scheduled to talk about the kings of the East tomorrow, and I will, but I'm going to do that relatively brief because I want to get at the challenge that we face in America. The most common question I get asked, two questions. Where's America in prophecy? I've read all the conjectures by various people. I don't accept any of them. We are conspicuous in our absence of mention, which couples with the question, why hasn't God judged America? I think he started. And why may shock you, and what's coming we need to talk about. And it's so important that I'm going to deviate from the announced schedule a little bit tomorrow. The Holy Spirit's really laid that on my heart. And I've really run way over time here, didn't I, Pastor? Yeah.
Stand with me. I know one thing. I know this is the smartest man in the room for sure, hands down. And I know another thing. I know he's the only prophecy preacher that could dare talk about glow-in-the-dark pigs. You are dismissed. God bless you.
Jesus likened his return to the earth as a time of sorrows and birth pains. This vital end-time message continues to go forth through the ministry of God's News as they host the annual International Prophecy Conference. This entire conference is available, along with many other prophetic materials, by contacting us. God's News Behind the News, Dr. Joe Van Covering, P.O. Box 10475, St. Petersburg, Florida 33733, or on the World Wide Web at www.godsnews.com. Stand with us as we proclaim this message. Jesus is coming soon. Be ready.